Good morning, students. Today we are going to discuss about the topic enzymes. This is the second unit which we have in our medicinal chemistry syllabus, that is enzymes and receptors. Receptors we have studied in our earlier classes. Now we are going to study about the enzymes. So in enzymes you have in the syllabus about the characteristics of enzymes. Second question is what are the factors that affect enzymatic action. Third one will be classification of enzymes. Fourth important topic is write about the kinetics of enzyme action, and fifth one will be the inhibitors of enzyme action. So these five concepts you have to focus on while learning enzymes and their action. So enzymes are nothing but biological catalyst. Okay, all the biochemical reactions are catalyzed by enzymes. So these are biological catalysts that speed up the rate of a biochemical reaction. Any given reaction, suppose it's a biochemical reaction, if it is in the absence of enzymes, it is taking 10 minutes, but in presence of enzymes, it takes only 2 minutes, okay, for the completion. So hence enzymes are used as catalyst in biochemical reactions. Most enzymes are proteins, okay, they are three-dimensional proteins. Enzymes are made up of proteins, okay. So these are the basic characteristics of enzymes. Enzymes are biological catalysts which speed up the reaction, and they are proteins, okay. The active site of the enzyme is the region where it binds to the substrate and it carries the reaction, okay. Active site is having a specific shape due to its tertiary structure of the protein, that we can see in the next figure. Enzyme action is very specific. Particular enzyme catalyze only particular biochemical reaction. So other reactions it will not influence, or it will have no effect. But on certain reactions only, certain enzymes, suppose lactase will be acting only on lactose, maltase will be acting only as maltose, but not on lactase. Like that, it will have specificity. Very specific reactions are catalyzed by the enzyme. So enzyme will have a active site. Active site means the site where the substrate binds to the enzyme as well as the catalysis is taking place. See here in the figure we can see that this is the enzymes. This total is the enzyme. This will have particular sites. This see this place and this place is called as binding site or active site, okay. Here in these places only the ligands or the substrates which have to undergo a reaction binds here, and thereby the reaction takes place. So these are called as the active sites or binding sites, okay. This total is called as enzyme. Enzyme is made up of two parts, that is called as cofactor and the inactive protein, okay. This total is a protein, and the cofactor is there, which is also a combination of amino acids, but without the cofactor the enzyme is inactive. Without the cofactor the protein is not active. This total enzyme is inactive. Cofactor binding activates the protein in order to act as the enzyme, okay. So there are various classification of cofactors that is not there in our syllabus. So let us move on to the next topic of our enzymatic reaction. So this is the enzyme. See, this enzyme is having particular structure, and this is the substrate on which this enzyme should show its action. So when enzyme combines with this substrate, here substrate goes and joins on the enzyme to form a substrate enzyme complex, okay. The reactant is called as the substrate, catalyst is called as the enzyme. Both together when they combine are called as enzyme substrate complex, okay. After the formation of the enzyme substrate complex, the enzyme helps in the lowering of the activation energy of that particular biochemical reaction. Thereby it enhances the rate of the reaction, okay. So where are the enzymes synthesized? Enzymes are synthesized on the ribosomes, which are further present on the endoplasmic reticulum in the cell. In the cell, endoplasmic reticulum is there. On that ribosomes are there. Ribosomes, we know that they help in the synthesis of the amino acids. They help in the synthesis of the protein from amino acids. So as enzymes are nothing but proteins, they are also synthesized in the cell on the ribosomes, okay. This is the point of synthesis of enzymes inside the cell. So next one is, what are the various factors that helps in the enzymatic action or that inhibit the enzymatic action? So majorly pH and the temperature are the major factors that influence the rate of enzymatic reaction, okay. So but this we will discuss later. But before going to that, we will have classification of enzymes. Okay, wait a minute. So we have till now studied what are the enzymatic reactions and how enzymes are synthesized in the body. What are the characteristics of enzymes? Enzymes are nothing but three-dimensional proteins. They are very much specific in their action, and they are nothing but combination of amino acids. Okay, so now let us move to the till now we have completed. Okay. So characteristics further, few more characteristics of enzymes are: enzymes speed up the reaction by lowering the activation energy. They does not involve in the reaction. They are again reproduced after the reaction. Their presence does not affect the nature and properties of the end product. Just promote the reaction, but they don't involve in the reaction. They are very specific in their actions. They are very sensitive to pH, temperature and substrate concentration. Okay, small amount of enzymes can also accelerate the chemical reactions. Okay, so all these are they will ask a question in the exam that write about the characteristics of enzymes. So 5 to 6 points you have to write. What are those? Enzymes are nothing but three-dimensional protein, those that enhance the rate of biochemical reactions. Second point is they are very sensitive to pH and temperature. Third point is they are very specific in their action. Fourth point is they enhance the rate of the reaction, but they don't involve in the reaction, so they are called as biocatalyst. Fifth one is they are synthesized in the cell in the endoplasmic reticulum. So if you write these 5 points, then you will have the full answer. Now, next one is a turnover number, which is in regard to enzymes. We know that turnover number is the number of substrate molecules transformed per minute by one enzyme molecule. Suppose one enzyme molecule is acting on 10 molecules of the substrate. Suppose lactase is there. Lactase converts 10 molecules of lactose to glucose and galactose. So the turnover number is 10. Okay, in that way turnover number of each enzyme is fixed. That many molecules of substrate it will act on. Okay, so nomenclature of enzymes. The enzyme is named based on the action which it shows. Okay, so majorly an enzyme according to the name of the substrate it catalyzes. Some enzymes were named before the like see if it acts on maltose, it is named as maltose. If it acts on lactose, it is named as lactose. Any enzyme in the end it will have ASE, A's or O's in the same. So by adding suffix like A's at the end of the name of the substrate, enzymes are generally named. Okay, so if it acts on galactose, it will galactase acts on galactose, maltase acts on maltose, lactose and lactase acts on lactose, like that. So whatever the substrate we choose, based on that only the enzymes names are given by giving this. See maltose here. So what is the enzyme name? Maltase. Like that every enzymes are named. You know about this very well. Okay, so these are some of the examples of the substrate, the enzymes and the products. Okay, so the substrate, if it is lactose, the enzyme name is lactase, and the products which we are getting is glucose and galactose. If it is maltose, maltase is the enzyme name and we get glucose from it. If it is cellulose, enzyme name is cellulase, and glucose, it is a polymer of glucose units. Okay, lipase, and it converts lipids into glycerol and fatty acids. Amylase converts the starch into maltose. Protease converts the proteins into peptides and polypeptides. We all know that. Okay, so these are the nomenclature of enzymes. Next going to the classification of enzymes. Okay, so these enzymes are majorly divided into 6 types based on the nature of action they are involved in. Okay, so this 6 types of classification of enzymes is given here as a table. So majorly they are of the following 6 types: oxidoreductases, transferases, hydrolases, lyases, isomerases and ligases. So oxidoreductases, what does these enzymes do? They involve in redox reactions. Redox reactions are those reactions in which oxidation and reduction takes place in a single reaction. Okay, so redox reactions, if they are involved, like lactase dehydrogenase, what does this do? This Dehydrogenase means it removes hydrogen from the lactose molecule. Okay, so oxidoreductases, they involve in oxidation reduction reactions. Next, transferases is one enzyme which transfer a particular chemical group from one substrate to the other substrate. These are called as transferases. Hydrolases is the enzymes which hydrolyzes. What type of bonds are hydrolyzed? Generally amides, esters. Okay, all those bonds. Bond cleavage with transfer of functional group of water. Okay, if water is involved and this water is added to two different functional groups after the cleavage. Okay, suppose if amide is there, what does the water do? It hydrolyzes and converts that into amine and carboxylic acid. Suppose ester is there, what does this addition of water do? It transforms that ester into carboxylic acid and the hydroxyl alcohol. Okay, in this way hydrolysis catalyzes the hydrolysis reactions of the enzymes. Okay, so next, this fourth class of enzymes are lyases. Lyases are those which breaks the bigger molecules to smaller molecules. Non-hydrolytic bond cleavage. Here hydrolysis is also a type of bond cleavage, but here whatever the 9 non-hydrolytic bond cleavages are there, that all comes under lyases. Lyases, or these are also called as lyases. Lyases means they break the bigger molecules to the smaller molecules. Next one is the isomerases. Suppose, see, we know what are isomers. They have the same molecular formula but differ in their orientation. So in the same way, here isomerases convert one type of isomer to the different type of isomer. Okay, so next, the final one is the ligases. Ligases are the enzymes which combine small molecules and to form a bigger molecule. Synthesis of new covalent bond between substrate using ATP hydrolysis. These are the ligases. Ligases are the enzymes which combine small fragments and make a big molecule, whereas lyases is the opposite. This breaks the bigger molecule into smaller molecule. Isomerases is the enzyme which helps in the isomerism, means like changing of the conformation of a particular substrate. Hydrolysis are the enzymes which helps in the hydrolysis of the some particular bonds in the substrate. Transferases are the enzymes which transfers a particular group from chemical group one substrate to other substrate. Oxidoreductases are the enzymes which helps in oxidation as well as reduction of the substrate in a single step. So this is about the classification and characteristics of enzymes. In the next class we will discuss about the factors affecting enzymatic action and the kinematics also. Okay, thank you.